Good afternoon, and thank you for the invitation. And thank you for, organizing this very nice meeting. So yes, today I would like to talk about projects we are currently running in the lab, and that aim at understanding how skin cells communicate, and especially how skin cells understand each other. And when you are looking just to this live cell movie, you can see that it's made of primary human melanocytes and keratinocytes. And clearly we can see that they try to touch each other to establish contact, they make projections, they are moving towards each other. So clearly there is a kind of dialogue that is established here, and they can somehow sense this dialogue. And when I'm preparing this kind of talk, it remembers me always very often, a movie that I like a lot that is called Lost in Translation. And I think today we have to slightly change the title, because it would be more how to not get lost in translation, and how these melanocytes and keratinocytes can succeed in understanding each other. So this work is performed by a very talented postdoc in my group, Leah Dominguez, and in tight collaboration with Clarence Research and Development team. You all know, I guess, that in the epidermis, so the outermost layer of the skin, or at least regarding pigmentation, you have two main cell types, melanocytes and keratinocyte. And this melanocyte here is really the melanin factory, so synthesizing the melanin pigment in a membrane-bound organelle called the melanosomes. And once this melanosome is fully mature, it will be transferred to keratinocyte in order to populate the epidermis and to give rise to skin pigmentation and photoprotection. And for instance, when the skin is exposed to UV, keratinocyte gets activated. They start to secrete several molecules like hormones, paracrine factors, as well as these extracellular vesicles called exosomes that we heard about this morning. And these factors are somehow able to lead to some transduction of signals inside the melanocyte. And one of the most famous one, I would say, is lead to the production of cyclic AMP, this second messenger, that is produced by the adenylyl cyclase. And this cyclic AMP is quite interesting in melanocyte, because it can lead to different phenotype, I would say, or it can lead to different processes, like pigmentation directly by promoting and the transcription of genes involved in melanin synthesis and melanosome biogenesis. But in melanocyte, and as in other cell types, the cyclic AMP can also help the change in plasticity of the cell. What I mean by plasticity is it can help the formation of dendrites and also the establishment of cell contacts. And of course, a cell able to pigment and able to make contact is likely, at least in skin, to promote the melanin transfer and to provide skin pigmentation. So this project aims at understanding how this melanocyte can receive some, or at least understand some extracellular signals, and to translate these signals into intracellular processes. So when we started this project, we, I have to say, have first a very naive approach. It was to, we decided to use human skin biopsy to perform electron microscopy on that tissue, in order to have a very close look at the ultrastructure of the melanocytes keratinocyte interface. The idea behind was that if there is some molecular platform, I would say, that are able to sense these extracellular signals and to translate these signals, it should be at this interface. So what we observe first is this kind of small membrane invagination here. I don't know if you see well. In blue, that is the plasma membrane of the melanocyte that is facing the plasma membrane of keratinocyte in red. Another view would be that one. So here we are in a melanocyte dendrite, full of this pigmented melanosome, and surrounding by this keratinocyte here. And you can see these vesicles, these small plasma membrane invagination here that are decorating the plasma membrane of the melanocyte. So we decided to have a better understanding of the morphology of this structure, and for that we used electron tomography. So this is an imaging approach that allow to reconstruct in 3D at the ultrastructural level the morphology of your sample of interest, I would say. So here we are still in the melanocyte dendrite. The plasma membrane is in green. The plasma membrane of the keratinocyte is in blue. And in red, you have the melanosomes full of pigment here, and you have this plasma membrane invagination here in white. So what we can see is that these plasma membrane invagination are still connected to the plasma membrane. Their diameter range between 60 and 80 nanometer, and they are non-coated. What I mean by coat is like a clattering coat, for instance. So this is really all the morphological features of one type of invagination that is called caveolae. So the caveolae are these schematized here, these plasma membrane invagination that are composed of different set of proteins, among which cavein and caveolin are really crucial and important, and among which the cavein one and the caveolin one are really crucial for biogenesis and function of this caveolae. Historically, they have been shown to control lipid homeostasis, like cholesterol balance, signaling events, like by controlling activity or by inhibiting signaling through GPCR, the G-protein coupled receptor, and more recently has been shown to control the mechanosensing of the cells, meaning that this invagination can be seen as a lipid reservoir that can flatten out or being invaginated. And by providing more membrane, more lipids to the plasma membrane, this membrane can cope with the mechanical stress and to avoid mechanical rupture, for instance. So in order to get to go a bit further in that examination, I would say, we decided to use synthetic skin. For the simple reason that we can monitor, for instance, the number of caveolae profile we can see over time of formation of the skin, because I guess you know that after seeding the cells, at day 4, the epidermis starts to stratify, day 6, it starts to pigment, and day 12, it's fully stratified and pigmented. So what we did is to monitor the number of caveolae profile we were observing at the two different interfaces we can see, meaning that the melanocyte keratinocyte 1, so the heterologous interface, and the homologous interface that is keratinocyte facing each other. And what we can observe is that the number of caveolae profile is much more enriched at this heterologous interface as compared to the homologous one. What we can do also is to quantify the contribution of each cells within that interface. And clearly, both cell types contribute to this caveolae at their plasma membrane, but what is interesting that over time of formation of the skin, the melanocyte is the one that will make evolving this number of caveolae, with a peak at day 6 where the skin starts to pigment. So to ease a bit our project, we decided to use a more simple system and to go in a 2D co-culture system. So what we did here is to use primary human melanocyte and keratinocyte, and we immunostain using antibody targeting caveolin 1 or cavein 1. So this is this white spot you can see here. And what you see is this linear pattern here at the plasma membrane of melanocyte that is facing the keratinocyte. So recapitulating somehow what we have observed in tissue. We can quantify this, and indeed, we have much more enrichment of this caveolae number at this plasma membrane of the melanocyte that is facing the keratinocyte. So since keratinocyte can secrete many factors and clearly have a very important role in controlling the physiology of the melanocyte. We decided to test whether keratinocyte might have a role in this amount of caveolae that are produced. So we use this co-culture system, but we also grow melanocyte alone or together with an irrelevant cells like HeLa cells, for instance. And what you can see here is this kind of polarization and intensity profile. And clearly, when melanocytes are grown together with keratinocyte, this enrichment and polarization of caveolae is much more increased or at least maintained. So since keratinocytes secrete factor, we also decided to test whether the secreted factor might have a role. And for that, we grow melanocyte in conditional media, keratinocyte conditional media. And here again, we observe an increase in this caveolae polarization. So meaning that together that the keratinocyte can secrete factor, and somehow these factors can tell to the melanocyte, okay, you have to maintain your caveolae. So since the melanocyte can also be stimulated by other means, like for instance UVB, we decided to stimulate melanocyte by low doses of UVB, and here we monitor the expression level of all the caveolae associated proteins, and they are all, their expression is increased over time, meaning very likely that the caveolae are much more formed and maintained at the level of melanocytes. So together, melanocytes can be stimulated, UV or keratinocyte, and this stimulation can maintain caveolae. So the question now is, what would be the role of this caveolae during pigmentation, for instance? So for that, we use siRNA to deplete the expression of caveolin 1 and caveolin 1. If caveolin 1 is not there, caveolae are not formed and not functional. And what we can observe quickly is that the expression level of tyrosinase, so one of the key enzymes for melanin production, is increased. This is associated with an increase in the production of pigment by the melanocyte, and also associated with more numerous pigmented melanosomes we can observe in cells. So together showing that caveolae are here to somehow restrict or slightly downregulate pigmentation in melanocyte. So the question is how caveolae can do that. So we decided to have a first look on the possible signaling events that would be under the control of caveolae. I remember you that indeed this keratinocyte can secrete factors. These factors, they can found their own receptor at the plasma membrane of the melanocyte, and this will lead to activation of signaling pathway, like activation of adenylyl cyclase that will lead to cyclic AMP production and lately pigmentation. So it has been shown in other cell types that caveolin 1 can interact with adenylyl cyclase and inhibit its activity. So we tested the hypothesis whether caveolae might negatively control the cyclic AMP production in melanocyte. For that, we use phospholipid to directly activate adenylyl cyclase, but by bypassing the need of having a receptor. So when we monitor the amount of cyclic AMP that is produced, so in control cells we observe upon phospholipid treatment a 4-fold increase. That is double when caveolin 1 is absent, so when caveolae are not formed. Showing you again here that caveolae can negatively regulate the cyclic AMP production in melanocyte. So we are here. Where caveolae can be somehow controlled by the keratinocyte and by stimulation of the melanocyte. And caveolae can also control signal transduction pathway like cyclic AMP production and pigmentation. So the obvious question for us is to test whether caveolae might have also a role on the cell plasticity. So our first approach was to grow melanocyte in different type of medium. The first medium is what we call a poor medium, meaning a medium where you have no factors that are susceptible to stimulate the melanocyte. What we can observe that in control or depleted caveolin 1 condition, that they are kind of elongated and almost indistinguishable from each other. When we grow them in rich media, meaning that we add some factors like, for instance, endothelin-1, I would say, the controlled melanocytes start to make some projection to adopt a star shape like, while the caveolin-1 depleted melanocyte is much more elongated and stretched, and this is exactly the same when melanocytes are grown in poor media but supplemented with forskolin. So here it shows that indeed caveolae can have a role on the cell shape and plasticity, and that this is likely under the control of cyclic AMP production because forskolin is not inducing any change here. So we monitor a bit more, at least we decide to quantify a bit more this observation. What we did first is to quantify what we call the aspect ratio, meaning that this is the ratio between the major axis of the cell and the minor axis. And what you can see is that you have an increase of this ratio when caveolin-1 is not present in rich media or media supplemented with forskolin. So meaning that indeed they are much more elongated and stretched. Another thing that we measure is the number of extension these melanocytes are doing. And when caveolin-1 is depleted and the cells are kind of stimulated, we are not able to promote several extension. More or less they are bipolar and looks like the control cells in poor media. So together showing that caveolae can have a role and have an impact on the melanocyte dendricity through likely cyclic AMP production. So the thing is now, okay, they have a different shape. So we decided to test whether cells can, this melanocyte can still interact with keratinocyte. So for that we decided to use live cell imaging in a co-culture setup. So primary human melanocyte and keratinocyte together, but melanocyte depleted or not for caveolin-1. And here you have the control melanocyte that can makes projection, that is very dynamic, that can also establish some contact with keratinocyte. And here the caveolin-1 depleted melanocyte that is elongated, stretched, polydynamic, and establish very few contact with the keratinocyte. So we quantified this by measuring the number of keratinocytes that are able to establish long-lasting contact with keratinocyte, long-lasting meaning at least more than 1 hour in our condition. And we observe a dramatic drop here of this number of contact, likely showing that this caveolae can stabilize this melanocyte-keratinocyte contact. So together, we are here. So we know that caveolae are polarized at the melanocyte in front of the keratinocyte, that it can control signaling pathway, pigmentation, plasticity, cell contact. So the obvious readout after that is to test the transfer, the melanin transfer in our condition. So the first thing we did is to use our co-culture setup. We immunostain melanin using antibodies that recognize the amyloid fibers onto which the melanin is deposited. And we quantify the number of keratinocytes positive for pigment and when they are positive, how much pigment they have. And these two parameters are decreased when caveolin-1 is depleted from melanocyte, suggesting that caveolae can contribute to melanin transfer in vitro. So then what I told you also is that keratinocyte can somehow control and enhance the amount of caveolae at the surface of melanocyte. So what we also decided to test is whether keratinocyte can do the opposite, meaning down-regulate caveolae. So for that, we came back in one of our previous studies a few years ago, where we showed that keratinocytes, exosomes secreted by keratinocyte, so this extracellular vesicle secreted by keratinocyte, can indeed modulate melanocyte pigmentation. So as you heard this morning, these exosomes are loaded with proteins, with lipid, as well as miRNA. And what we identified, at least in that study, that one miRNA was loaded in that exosome was able to modulate pigmentation of the melanocyte. And interestingly, the same year, the group of Lionel Larue showed that that particular mRNA was able to target caveolin 1 in melanoma cell. So what we decided to test is whether that mRNA can also have a role through caveolin on the transfer of pigment. So this mRNA is called 203A. We express it in melanocyte. Indeed, the caveolin 1 is downregulated. And as a consequence, we observe that the tyrosinase is increased, as we showed, for instance, in the sRNA condition. And also, using this co-culture setup, we observe a drop in the amount of pigment that is transferred from melanocytes to keratinocyte. So here showing that if indeed the caveolin can contribute to melanin transfer, and potentially the keratinocyte might be able to also control that event. So now the thing is that, okay, we are in 2D, so we would like to possibly test whether what I'm telling you is also true in 3D. So for that, we had the chance to collaborate with Clarence and to have Nathalie André, who is in the audience today, who has a know-how, how to generate this synthetic skin. So what she did is to use primary human keratinocytes together with control melanocyte or caveolin 1 depleted melanocyte. And then we decided to analyze this tissue at the electron microscopy level. So here we are in the control epidermis. You have a melanocyte control here, full of black pigment, and the neighboring keratinocyte here are also pigmented, illustrating that the transfer has occurred. At some occasion, we can also observe this naked pigment here that is likely secreted by the melanocyte and ready to be uptaken by the keratinocyte. So then, when we go in the caveolin 1 depleted epidermis, this melanocyte is still pigmented, but this is coherent with what we showed in vitro, in 2D. However, the neighboring keratinocytes here are devoid of any pigment. So we quantify this observation and indeed observe a decrease in the number of keratinocytes that are positive for pigment, and once they are positive, they have less pigment in their cytosol. So showing that caveolin contributes also to melanin transfer in 3D and in synthetic skin. So now I would like to conclude and try to leave you with this kind of idea that caveolin is able somehow to organize spatially the melanocyte. I mean, to tell the melanocyte what to do and where. Because indeed caveolae are polarized in some area of the plasma membrane of the melanocyte, meaning that some areas are kind of devoid of caveolae. So if we speculate a bit, we can imagine that some signal transduction might be more activated in some area of the cells or more downregulated in some others. And the same would be true for these dendrites and plasma membrane plasticity. So I think it's really important for polarizing the melanocyte, and on top of that, the keratinocyte is able to control these features either positively or negatively. And I don't think it's a pure on and off system. I would like just to leave you with the idea that I guess it's a slightly on, slightly off process, and that the fine-tuning of this amount of caveolae, or the fine-tuning or where you will position this caveolae, will be likely important for the overall pigmentation of the tissue. So with that, I would like to thank all the people working in my group, especially Lilia Dominguez, who performed that work, the team of Graça Raposo, in which I'm working on, Issue Urbain, Maryse Romao, Xavier Guilchen, and Florence Passin, who perform a lot of work on the electron microscopy. I would like to thank the group of Christophe Lamas at the Institut Curie, who is an expert in caveolae. So it's really helpful because I'm not at all an expert in caveolae. And also, I would like to warmly thank the research and development team of Clarins, so Catelvier, Christelle Guéret, and obviously Nathalie André. The imaging facility at Institut Curie, our funding, and you for your attention. So thank you so much.
Thank you very much, Cedric, for this very nice presentation. So the discussion is now open. Do you have some question in the audience? Yes, Marek? Just at the front.
Thank you very much. I always look forward to your speeches because it's so clear-cut and well illustrated. Now, electron microscopy is a fine approach, but you have to be lucky to spot the right moments that happen in the skin. It's, however, somehow puzzling that so rare are the pictures when you can see really the pigment transfer. You show some melanosomes outside of the cells, but the intermediate steps are very rare, anyhow, rarely observed. Do you have any idea why? But because
I can just give you my guess.
Yes.
But it's true that it's very rare. It's slightly, we can observe slightly more often in synthetic skin, I have to say, than in human skin biopsies, for instance. So my opinion is that the intercellular space between the melanocyte and keratinocyte is very narrow. And I have this, the keratinocyte is known to be almost kind of a phagocytic cells. So I really believe that once a pigment is outside, it is very rapidly eaten by the keratinocyte. At least what we observe in vitro, because we have also another project on that, clearly when you feed a keratinocyte with pigment, very rapidly the pigment is inside. So in skin, of course, I don't know. The thing is that also in skin, you are in a kind of steady state situation. It would be, I would say, maybe better to potentially do some electron microscopy on skin that have been just exposed to UV. Well, I mean, this is doable. We didn't do it, but this is doable. But I really think this is that, because there is so few space in between the two cells that once it's there, it is just catch.
So, well, anyhow, there is no direct transfer of pigment apparently from cell to cell. First, there is an intermediate step of secreting melanosomes into extracellular spaces, apparently.
So there is many different hypotheses that has been published in the literature. The only thing I can say is that in our hands, when we are manipulating either human skin biopsies or human synthetic skin, the only events we observe is secretion. I never observe like the nanotubes, I never observe like phagocytosis of a dendrite or whatever. So it's not because I don't see that it's not existing, but based on what I have observed, I would say that in human skin, the major process should be, in my opinion, the secretion.
Yes, it's a quick question. Oh.
Okay, perhaps it is a naive question because I do not know much about that, but is the part of the membrane in the melanocyte which is enriched in caveolae, is it similar to lipid raft? I mean, enriched in sphingomyelin, cholesterol receptor, and also is it the part of the membrane where the melanosome can go out of the cell?
Naive, but very good question, I have to say. Caveolae are known to be enriched in cholesterol. So even if we didn't check carefully, I have to say, it's most likely to be the case. Regarding the secretion part, I don't have clear answer because we tried to have a look to that. The thing is that when caveolae are present, at least the area where caveolae are not the area of the plasma membrane that will be the one that will make these dendrites, for instance, this protrusion. So this part is more rigid. So my feeling is that if secretion occurs somewhere, it should be on the other part. I have no real clear evidence for that because it's almost impossible to follow the secretion in vitro like this, but this is my guess.
The other Corinne?
Cedric, do you have any details on some potential anchor protein anchor to the caveola that could, and eventually the receptor on keratinocytes that would be involved in the stabilization between, in the dialogue between melanocyte and keratinocyte, and that would contribute to the transfer of melanosomes?
If I had the answer, I would have presented, But no, so the caveolae are known to be anchored to the actin cytoskeleton. So, and we really believe that the way it is anchored to the cytoskeleton or how it can possibly remodel the cytoskeleton would explain some of the phenotype we are regarding the cell plasticity. Regarding the receptor, we have absolutely no idea. What is known in other cell systems is that several J protein coupled receptors are found in caveolae. It's still not clear whether they are there to be inhibited or to be activated. So again, my guess would be that, I don't know, endothelin receptors, MC1R, this kind of guy should be somehow found close, nearby, or within the caveolae. To be able to regulate the activity. So we tried a bit in that direction, but not too strong. So I cannot tell you more than that.
Is there any last question? Yes, Frédéric.
You mentioned an article of Lionel Larue on P10, and because in patients with P10 with germline P10 mutation like Cowden disease, you have l'antigen in buccal area and in genitals, and you also have an increase of melanoma. So I was wondering if the abnormality of pigmentation in this disease is related to caveolin, and if P10 is located in the membrane of caveolin.
The short answer would be, I don't know. But the thing is that caveolae have been shown, there is many studies about that, to be either a tumor suppressor or a pro-tumorigenic factor. So I think it really depends on what are the downstream effectors. It can be very different from cell to cells or depending on the context. So to be honest, I don't know. Regarding where is P10, I have absolutely no idea.
Because normally it's just under the membrane, in contact to phosphatidylinositol 3 phosphate, and so the location is just under the membrane, and but I don't know if there is an enrichment in special area like caveolin.
I don't know either, but if there is any link between association to membrane that would be kind of enriched for cholesterol, thymomelanin, and all these things, it would be very likely. But to be honest, I don't know.
Okay.
Thank you.
Thank you very much.
Thank you.